So in the wake of the pandemic, the research landscape has undergone a dramatic shift, and at LSTM, we've been at the forefront of this change. We've seen a renewed commitment to understanding and combating high-consequence pathogens, in particular viruses, that pose direct and growing risk to public health. As a result, LSTM researchers have turned their focus to addressing the most urgent challenges in diagnostics, therapeutics, and viral biology. The next series of talks, the short talks, will give you some examples of these activities. So without further ado, can I invite Ana to come up? Ana will begin with an overview of the challenges and recent advances in diagnosis of Crimean-Congo Haemorrhagic Fever.
Thank you. Thank you, everyone, to stay past 5 p.m. To listen to the talk, and thank you very much as well for the invitation to be here today presenting. So thanks, Giancarlo, for the introduction. I will start talking first a little bit of background on Crimean-Congo Haemorrhagic Fever, or CCHF. CCHF is a virus of the Bovine-derived family. It's transmitted by a bite from a tick of the genus Ialoma, and as well as by contact with body fluids of infected humans and animals. Symptoms are non-specific of CCHF, and these start by similar to flu-like symptoms, and these follow hemorrhagic symptoms, such as, for example, petechia, red eyes, areas of bruising, and then in severe patients, we see internal bleeding, organ failure, and death. CCHF has been reported in more than 30 countries and is endemic in many countries in Africa, Central Asia, and Europe. There has been an increased incidence in the recent years in Turkey, Iran, Iraq, India, Greece, Georgia, and Kosovo, with the addition of the appearance of new countries such as Spain and France. CCHF is a WHO priority disease. There is no licensed treatments, there is no vaccine, and has a high fatality rate, between 10 and 40%, but in limited outbreaks, the fatality rate can be as high as 80%. Diagnostics is very challenging. For CCHF is slow. Currently, samples are sent to reference laboratories for testing, and this has a delay of obtaining the results between 2 and 7 days, depending on where the laboratory is. This is impeding the appropriate management of patients and treatment. So because of this, the WHO has prioritized the development of point-of-care diagnostics or near patients rapid diagnostics for this disease to speed up diagnostics. An additional challenge for CCHF is that a biosafety pathogen, where it makes very difficult to evaluate new diagnostics because there are not many laboratories in the world that have this high level of containment. So to overcome these challenges, we have been working on different programs at LSTM. So the first one is the development of a point-of-care molecular diagnostic test for CCHF. This has been done in collaboration with a company called the GeneDrive, which is based in Manchester. The second one is the development of a rapid diagnostic test for detection of CCHF antigens, and it has been done in collaboration with a company based in Bedford called Global Access Diagnostics. These two projects try to fulfill the gap of development of a point-of-care diagnostics or rapid diagnostic test for CCHF, which is a priority for the WHO. And then the third one is the development of a CCHF pseudovirus, which has also been prioritized as research by the WHO. So this is the GeneDrive platform. You can find one in the stand outside for the Center for Drugs and Diagnostics. It's a portable PCR platform, approximately of this size. And then, we developed a PCR assay to be used with this platform, so we could use it at the point of care. The GeneDrive platform is small, it's portable, it's lightweight, it's commercially available, and is CE mark. It has a simplified sample prep. It doesn't require RNA extraction, so simply you add the serum sample in the cartridge, press the button, and then it runs a PCR, and then it tells you in the screen if it's positive, negative. You can also connect it to the laptop to get the raw data. So pilot testing of this assay was performed in UKHSA among 150 serum samples, and we obtained a sensitivity of 94.4 to 96%, which is very good. So following this evaluation, we freeze-dried the assay. By freeze-drying the assay, it not only facilitates transport and storage, but it also brings the test closer to the point of care, which is the ultimate aim of this. So we evaluated the freeze-dry assay at the Ministry of Health in Ankara, and then we obtained comparable sensitivity and specificity with the wet assay. Here is just an example of the dry freeze-dry cartridge. They're also outside if you want to have a look in the stand. The second project is the development of a lateral flow assay for detection of Crimean-Congo Haemorrhagic Fever antigens, in this case against the nucleoprotein antigen. So these are the steps that we follow to develop an RDT. First, because we are detecting antigens, we develop monoclonal antibodies. We selected best antibody clones for this. We develop RDT prototypes through several rounds of optimization and testing. We led to a lead prototype, which was evaluated last year in Turkey and Iraq. So the sensitivity and specificity that was obtained among 125 prospectively recruited patients in Turkey was 90.4 and 96.2. This sensitivity and specificity fulfills the WHO target product profile for CCHF RDTs. Sensitivity in Iraq was slightly lower. Following this evaluation, we obtained further funding from the MRC DPFS award to take the prototype to the next stages of development. Here, we have a diagram of the different phases of development of a device. We have already passed phase 2, which is proof of concept, and phase 1, feasibility. We are currently in phase 2, which is the final optimization. And then with this program, we want to get to launch. So currently, we are in phase 2. We have done another round of optimization of the device to increase sensitivity. And currently, it's being evaluated through our trial DETECT in Iraq and Turkey with a larger sample size. We are expecting to finalize this phase at the end of this month. Then we will start phase 3, which is technology transfer and upscale manufacturing of the device, which will end up in the sign lock of the device. During technology transfer, we look at what is the best conditions for manufacturing the device at high volumes, where we are still preserving the original performance of the device. And at the end of this phase, the device will be design locked with all the kit components. Then, once the device is design locked, we are planning to evaluate it in the field next year at the point of care in 6 clinics in Turkey. We will also carry out an initial of use and acceptability study for information on future implementation studies, and we will carry out a matrix equivalency study to evaluate the sensitivity and specificity of the device in serum, whole blood, finger prick blood, and as well using this TASO device that is a self-contained biosafe capillary collection device. And finally, so the aim is, following this evaluation, to build a dossier for regulatory approval of the device. And then finally, at the end of the program, we will plan to launch the device upon obtention of the regulatory approval. And then finally, so we are as well developing a pseudovirus for CCHF to express glycoprotein to use as proxy for CCHF virus, but to be able to perform the experiments in biosafety level 2 conditions. This will enable carry out, for example, measuring neutralizing antibodies, viral entry experiments, and the aim is to take this for implementation in endemic countries for capacity strengthening. Recently, the WHO Task Force for Viral Hemorrhagic Fevers, this is the last slide, met to set the priorities for the next 6 years. The priorities continue being the development of commercially available point-of-care tests, and rapid diagnostic tests continue being a priority. With these projects, we hope that we can make a difference to get closer to this goal. It was highlighted the importance of integration One Health approach and vector surveillance system is in CHF endemic countries where currently is not performed. CCHF is a disease of the rural. High-risk groups are farmers and abattoir workers. And, for example, in Turkey, 90% of the infected people are farmers. So One Health approach for this disease is key. Something that also needs to be done is to update the WHO target product profile to include point-of-care molecular diagnostics. We obtain very good results with the gene drive, but there is no threshold of sensitivity and specificity set in the target product profile, so we don't have any reference to refer to progress. And then finally, develop biobanks, networks with countries' repositories of well-characterized samples to facilitate the evaluation of novel diagnostics. And then I would like to thank a big team of LSTM investigators, especially Kaylee Thompson for the RDT prototype, Tom Fletcher, Thomas Edwards for the Gene Drive project, Christine Goffinet for her help on the pseudovirus system. I would like also to thank the Global Health Trial Unit that are doing an amazing work on the DETECT trial. Our partners, commercial partners, Global Access Diagnostics, Gene Drive, and of course, our in-country partners that couldn't do anything without them. Thank you very much. Questions or?
Thank you, Anna. If I can invite Alain, in the meantime.
Okay.
Is there a quick question for Anna? And Alain, right, if you can set up. Any questions out there? Anything online? Well, it's an amazing achievement, so well done. Thank you very much. Okay, next up is Alain, who's going to take us through the intricacies between arboviruses and the vector host.
Okay, hi everyone. I'll just wait for my slides to come. Yep, here we go.
Hard. It's hard.
Harder. There we go. Okay. Got there in the end. Well, thanks everybody. I'd like to especially thank all the organizers. In particular, Mark, I don't know where he is, but as an organizer myself, I know how much work it is to put something like this together. And what I'm going to tell you about is work that started at the end of 2007, so quite a while ago. It's essentially just to sort of tell you a little bit about the journey. I don't have a great deal of time to give you any of the detail, and I apologize to my colleagues who are clinicians and public health people, because you'll probably find this really, really terrible. But the point is also not to understand the details. It's to show you a little bit about the sort of work that that is going on, the diversity of work that's going on at LSTM that stretches from the fundamental to the sort of applied science. And any applied science starts usually with something fundamental. Okay. Right. So I want to start by thanking but it's really sort of the most important people who've been involved in this. And there's been probably 40 or 50 people over the years. I can't put all of them on here, but I want to highlight at least some of them. That was from, which is what essentially this work started and was continued, not at LSTM, but from the past. I would like to highlight Esther Schnettler and Margus Vajak, who are now professors in their own right, who are absolutely instrumental in bringing this work forward. And there are a number of key collaborators, and I really just want to point out Rhys Parry and Anders Meritz, who've been really, really great over the years. And if I can give a piece of advice to my younger colleagues, collaborate a lot, collaborate wisely, and you'll be a lot more productive than you'd be otherwise. Right, okay. I don't think I have to explain to this audience what arboviruses are. I think most of you will at least have a vague idea of what's going on in these, with these pathogens. Just very briefly, okay, what I want to point out is that arboviruses are transmitted by arthropods. It kind of comes with the name, like mosquitoes, midges, ticks, and so on, to vertebrates. We have these transmission cycles that can be simple, sometimes more complex. The point I really want to make is that arboviruses replicate in vectors, i.e. arthropods, and vertebrates. And when you have replication, what you have pretty much throughout nature, you will have detection of pathogens, you'll have induction of host responses, immune responses. And that is something that's really commonly found everywhere, and that's something that's really important, because what I want to explain to you today about why do we do RNAi, why we're interested in this, is because we want to see, well, can we understand what's going on in vectors in order to prevent this step here, transmission. So you've heard about vaccines and so on, very important, antivirals also very important, but breaking the transmission cycle is a different part of the step. And it's very rare, I think, that these problems are solved with one approach. Okay, so I think that's why there's been a lot of investment, like scientific time, thinking, money into understanding, like how arboviruses interact with vectors. Right, so over the years, my group has been working on lots of different arboviruses, be they transmitted by, this is one odd pointer, man. It's like a wonky lightsaber. We've been working on mosquito, tick, sandfly-borne arboviruses. Some of them are listed here, so Bunya viruses, flaviviruses, toga viruses. And the details here, the molecular details, aren't really that important. Right? You can essentially forget all about this. What I want to tell you is that we've been working on these. There's lots of tools for those viruses. There's important human pathogens, animal pathogens in all these groups here. We've been working a lot on Seminichiform virus, so this one here, which is a close relative of chikungunya, for a number of experimental reasons. It's a great virus to work with and understand RNAi, but we've been using pretty much all of those other viruses here in these different systems as well. Okay, again, this is something I don't think I need to explain to this audience, but the point I really want to make, which is sometimes not that obvious to the public, I guess, is that, yes, there are different types of mosquitoes. There's Anopheles, there's Culex, there's different types of Aedes mosquitoes. There's, of course, differences between all these mosquitoes and how they behave, what they do. Are they diurnal, are they nocturnal, and so on. So there's people in LSTM who try to understand that, of course. And this is really, really important in understanding how you fight mosquitoes, how you fight how a virus or parasites transmitted. This is really important. We've been mainly working on, for a number of reasons that we can discuss at a different time, we've been working on Aedes aegypti, the yellow fever mosquito, which is so critical for many different arboviruses. So that's the one we've been focusing on the mosquito side for well over a decade. Okay, this is where things get a little bit molecular, and again, the details are not really that important, right? You don't need to understand everything or every inside out I'm going to tell you. What I want to appreciate is that this started a long time ago, right? And some of you may think, oh, well, you know, RNAi, we've seen these things before, we understand this. Didn't really understand it in 2007, okay? There were lots of gaps, and that's when we sort of started working on this. There were a lot of steps of this pathway that weren't really that clear. Sorry, that weren't that clear. So what we believed at the time, and this is effectively a slide I made a long time ago, is that, viral double-stranded RNA, so when viruses replicate, they make double-stranded RNA, that's a replication intermediate, is chopped up in mosquito cells, right? So that's all in cells, by a molecule called DISR2 into small RNAs. And so we call these VS RNAs. We had a good idea from the Drosophila field that they'd be looking like this somehow. So 19 nucleotide overlap and then 2 unpaired bases at each end. So 21 nucleotides for each strand. So that was sort of the going measure at the time. And what would happen is that when you have these small RNAs, there's a molecule called Argonaute 2, which retains one of the strands, so what we call the guide strand, which is used to find complementary RNA. And if you find complementary RNA, then we presume we would have cleavage events. And if you degrade viral RNA, of course, you'll stop virus replication, right? So that was sort of the theory at the time, and what you can show in infected cells. So these kind of red dots here in mosquito cells. So these are double-stranded RNAs, right? So you can detect them very, very, very easily. So that's really not a problem. So we know that the substrate's there. Viruses replicate, they produce the substrate. And if you wonder, well, how generally relevant is this? If you're in a human cell or an animal cell of any sort, that's exactly the same thing that happens as well. That's how cells detect viruses, by detecting double-stranded RNA mostly. So that's whether you're an arthropod or human, be very, very similar. Okay, so we didn't really know whether any of this is actually true and how does it actually really work. And I don't want to bore you too much with the details here either. But what I'd like to point out again is if you're working on mosquito cells, you're about 30 years behind what's happening in human cells. So we had to get to the point, and that's a colleague of mine at Pirbright called Karen Maringer, who developed knockout cell lines. So they're all derived from an Aedes aegypti cell line, which is very widely used in the field. So we have DISR2 knockout cell lines, AGO2 knockout cell lines, and you can use that to reconstitute expression with a new generation of plasmids that we made. So that was a big change in the field, that we were able to actually express proteins in mosquito cells. The first attempts that I, to some extent myself, made in the early 2000s were pretty pitiful, I have to say. We now have much, much better ways of working with mosquito cells. So with all these sort of technical advances, of course, you can then, you can really investigate what's going on. And again, this is really just one approach of how you can show that something is important or not important. So if you infect cells like this, so these are DISR2 or AGO2 knockout cell lines, we infect them. So this is 74 virus, the genome, so we put a reporter gene in there, luciferase. The idea is essentially, if you get more luciferase, right, you have more virus replication. So it's a fairly simple thing. And if you put that on cells, and there's different people have done this differently, this type of experiment, this is really just to show you how we did it at some point. If you compare to like a parent cell line, which has a functional siRNA pathway, and you knock out DISR2 or AGO2, well, you get a lot more virus replication. And that is one way of demonstrating that this particular pathway is actually important. So, yeah, well, that's a good demonstration. I said there's others, but it's one way of doing it. So we then went on to understand or try to understand how these molecules actually work, what do they do, and the details here are completely unimportant. I just want to show you what you can do when you have these tools at your disposal. So you can do these kind of molecular studies with structures, and you can understand, you can look at the different domains of a molecule, such as DISR2. So here it is, here's a DISR2 structure bound to double-stranded RNA. And you can understand, well, what do these domains do? So they have a helicase domain here that actually binds to double-stranded RNA, an RNase domains that cleave double-stranded RNA, a PAS domain here that holds everything in place. And you can relate sequence to structure. You can relate sequence to structure in a really, really nice manner. And that's one way of understanding, really, what's going on and how these molecules actually act. So I think that's... for us, at least, that's a major, major step forward, and I could say considerable progress for the field. You can do the same thing with Argonaute 2, so another protein, obviously, that I just mentioned. And again, what we can do is we can look at the structures of these proteins, we can analyze the structures. And for those of you who are interested in Argonaute proteins, and I might well be the only person in this room who's interested in Argonaute structures or function, but nonetheless, not all Argonautes have cleavage activities, so they can work as RNases, but they don't always do. And so what we did, for example, is we identified catalytic tetrads, so like the enzymatic activity in Argonaute proteins, and what this actually means for inhibiting virus replication. Okay, so you can really use that to understand how Argonauts work, what do they do, and so on. So yeah, we are now at a point where when we're looking at this specific siRNA pathway, which is the exogenous pathway, but the details again aren't really that important, we're now at a point where we can sort of like tick off this slide and say, well, this is where we are, right? We have a fairly good understanding of how this works, so now we can use it. And so while we are more on the cellular side of things, on the last year, or this year and the last year, a number of colleagues who are sort of real entomologists have made knockout mosquitoes. And so what these guys find is that, yes, if you take away this pathway, if you take away this host response, what you see is that these mosquitoes are really, really unwell when you put viruses in there. So it's a nice coming together of understanding what's happening in a cell as to what's happening in vivo. So I think we're now at a point where we can say, well, we understand how this pathway works, we understand it's important, and we can now move forward from there. So what you can do with this, well, you know, what have these many, many years of working on mosquitoes actually taught us? Well, we can, for the different viruses like flaviviruses, togaviruses, bunyaviruses, we understand the nature of small RNAs, we understand how they're being produced, we understand sort of like, to some extent, the finesse between the different types of interactions. And there's other pathways as well that are important that I don't really have time, but we've done a lot more on this. But what's cool actually in a way is that you can use all this information and everything that you've learned, you can move into other vectors like sandflies and ticks. So that's what we've done. So we now have, I think, a pretty good understanding of what's going on. And now we have colleagues who, which is not me, we're colleagues who are proper mosquito geneticists, who are using this information to produce transgenic mosquitoes that we hope will be able to sort of inhibit transmission. That works in the lab to some extent already. Hopefully that will be usable in the field as well at some point. So I think that much of this work has been useful. Just want to, you've seen this before, right, Mark? I thought I would finish off here. What can you do with all this RNAi stuff? Okay, what can you go from one invertebrate to another invertebrate? And I really just want to sort of show again what Mark had sort of introduced already on Brugia millei, this parasitic nematode. And one of the viruses which is related to a chikungunya virus called it's the wrong one. I always get these viruses, BMRV1, not OVRV1. Anyway, so if you want to understand how these viruses and these worms interact, so these are viruses here labeled in worms. Well, you can use RNAi. So you can use all the technology and all the scripts that have written, all the bioinformatic tools to see whether these viruses are replicating or not, and you can do all this very sophisticated analysis, which nowadays is actually fairly standard because we've done it so many times, to move from one to the other, and it's very neat, nice technology transfer in many ways. And yeah, I just want to leave it here. So thank you for your attention.
Thank you, Alain. Is there a quick question for Alain, as Christine? No, well, thank you very much, Alain. Okay, next up is Professor Christine Goffinet, who will take us through her journey in exploring HIV cure strategies, in particular the shock and kill.
Yeah, thanks, Giancarlo. So sadly, I didn't make it to be a speaker in the best session, which was reported to be yesterday. So I will do my best to continue to speak about HIV-1. However, really shifting gears now, going back to the really begin of the translational pipeline and talking a bit about efforts to advance HIV-1 cure strategies. So I don't need to introduce HIV-1 to this audience. We are benefiting from excellent antiretroviral therapy options. I think HIV-1 is the virus with the best and largest portfolio of antiviral drugs. However, this comes with a caveat. This antiretroviral therapy is not curative. So we have here the numbers which have been iterated yesterday. We have still huge numbers of people living with HIV-1 today, almost 40 million, and last year 1.3 million estimated individuals have acquired newly HIV-1. So the best case scenario for these people is to access antiretroviral therapy under good conditions and a good medical, being accompanied by good medical support. However, this comes with development of resistance, side effects, a huge problem with stigmatization, which again has an impact on access and adherence. So the holy grail of HIV-1 research, in addition to finding a vaccine, is actually to advance and find a strategy for HIV-1 cure. And also, if you listen into communities of people living with HIV-1, and I like to read here this very famous anonymous quote, is that you will hear, one day I would love to say, I used to have HIV. So we think it's a really important scientific goal. This is an urgent and important clinical need. However, we have to confess we are quite clueless where to start. So why is actually HIV-1 infection a chronic and persistent disease? It is actually a quite clever pathogen. It has evolved to integrate its genetic information into the chromosome of the human cell it infects, so making this persistent and fixed basically forever. And this proviral DNA is not always transcriptionally active, and this is exactly the trick. It's only sporadically active. And this results by a high proportion of HIV-1 infected cells being actually transcriptionally silent or dormant. So it would have been an easy task if silent cells or cells expressing a silent HIV-1 proviral DNA integrated into the chromosome expressed one or several specific biomarkers, so cell surface proteins, which would actually label these cells as being HIV-1 infected. Then we would design a very targeted strategy that would target this specific proportion of cells, maybe coupled to a toxin or whatever, and it would be probably quite easy to eliminate this specific population of cells. However, this biomarker has not been identified and it might not exist. So the HIV-1 infected cells in the absence of viral replication might just look like every other T cell of a specific subset. So the field has moved on, and there was quite some excitement, I think 10 years or so ago, when researchers figured out that shock and kill could be a valuable approach to tackle this scientific question. So rather than accepting that the viral proviral DNA is transcriptionally silent, let's force the virus to express genes to make proteins which would then make or render the cell immunologically visible. So this first step would be a shock intervention which would activate viral transcription. This should then lead to activation of transcription, proteins would be made, and especially we would have surface expression of the only protein made by HIV-1 which is located on the surface, which is the envelope glycoprotein. So this will be important for the rest of my talk, which is this little guy here, the trimers expressed on the surface of these reactivating cells. And in a second intervention, or let's assume the immunological system of the person itself should now be able to eliminate these cells, because these cells are now visible for the immune system. And it goes without saying, this strategy only makes sense if we couple this or include an antiretroviral treatment, because otherwise, of course, the newly produced viruses would initiate new rounds of infection. So that's a very nice textbook and idea or concept. This has not worked in the clinics. People see nice reactivation induced by specific latency reverting agents, and there's a myriad of compounds which have been tested, but the reservoir, so the number of cells carrying an HIV-1 integrated DNA, is not really significantly shrinking, indicating that we need to better understand what's going on. So again, this starts with the proviral integration site and sequence. A good latency reverting agent should then initiate viral transcription. We then want to have glycoprotein surface presentation, because this labels the cells now as HIV-1 positive. But then, in addition, immune killing mechanisms must come into place, antibody or T cell mediated. So to tackle these issues, we decided to generate a novel reporter virus to make our life easy to detect envelope glycoproteins. So it's very simple. We actually inserted a GFP sequence in a variable loop of the envelope glycoprotein. So this results in reactivating cells looking like this from the exterior, where you have ENV trimers being accessible from outside. And in addition, they will express, or this will be including a GFP molecule. So for those who are familiar with flow cytometry, this really allows us to not measure reactivation per se by measuring GFP fluorescence, but in the same sample using anti-GFP antibodies, quantify the accessible envelope glycoprotein on the surface. So we started with some prototypic latency reverting agents. The names and the mechanisms is not so important today. And we started by measuring in a titration experiment the ability of this drug to reactivate HIV-1 protein expression by measuring the quantity of the most abundant HIV-1 antigen, which is P24, and expressed inside the cells. So you see that bryostatin is a quite nice and good LRA, and JQ1 is clearly inferior. However, when we looked into more detail and looked at envelope glycoprotein expression, we noticed very quickly that bryostatin was actually inducing an interference with the synthesis and the cell surface presentation of this important trimeric structure, which we want to be expressed for elimination. When we added those two drugs together, we were observing the already reported synergistic effects of these two drugs. So we thought, well, maybe actually this ENV defect is not being repaired, and this might be actually a good strategy for shock and kill. And this is shown here again. Envelope cell surface levels are quite bad for JQ1 because it's a quite poor LRA. Bryostatin induces poor levels because it seems to counteract ENV expression. But when you put two together, you get very impressive ENV surface levels. And you would have expected that this now directly translates in a good ability of the immune system to eliminate these cells, right? But that's not the case. Very surprisingly, this double combination behaved very poorly in a antibody-dependent cytotoxic killing assay, which really indicates that the mere amount of ENV on the surface does not predict the ability of these cells to be killed. So we had many hypotheses what could be going on, and one of them was whether this double combination was actually somehow changing the intracellular state of these cells and rendering them maybe more resistant to killing. So we decided to shift gears and include now let's call it an apoptosis facilitator, which has also been described as an LRA, and to see whether we could actually render these cells more susceptible to killing. So this is just to show again the reactivation by the drugs are already introduced to you. AZD, the apoptosis facilitator, is actually a quite poor LRA, but when you combine it with JQ1, again, we see really great reactivation, almost as good as the Bryo-JQ1 combination I introduced earlier. By conducting an experimental apoptosis experiment, so by using an inducer of apoptosis, we actually confirmed our suspicion that bryostatin is clearly interfering or inhibiting the ability of the cells to react to an apoptosis inducer. And as expected for an apoptosis facilitator, this was not the case for AZD. So you might now think, that's maybe a perfect combination, right? We have great reactivation, we have great ENV surface levels, and we have the full susceptibility of the cells to react to an apoptosis inducer. Well, if I'm asking like this, maybe not, because we didn't integrate the contribution of the virus itself. When you look very carefully by flow cytometry, you can actually measure apoptosis in cells which reactivated and in those who stayed silent. And it's very clear that those who reactivated, so which made antiviral proteins, clearly showed a reduction in the ability to induce apoptosis. So we have a gene product expressed by the virus, which is reactivating, which is forcing the cells to be more resistant to apoptosis, which, from the perspective of the virus, of course, makes a lot of sense. Lessons learned. Dylan Posmus generated this work, and actually this is just a fraction of the data he generated, and I have to say it's a really fantastic PhD student. Watch out for him. He will be on the postdoc market in a year or so. And the second lesson learned is the cure for HIV-1 is not around the corner. And I think me as a fundamental scientist have to say researchers, but also media must be very careful in not over-promising or over-hyping initial results which might look promising however, which do not translate into something clinically applicable. The mere reactivation of HIV-1 is insufficient to shrink the virus reservoir. LRAs may very well be capable to reactivate HIV-1, but some of them might interfere with the second step, which is urgently needed. The virus itself works against a shock and kill approach, and we haven't even started thinking about the importance of the sequence of the HIV-1 provirus, different subtypes, the integration site, and the different susceptibility of subsets of T cells and monocytes to react to individual LRAs. So there's still a lot of work to be done. So Dylan is not on his own. He is in a wonderful team, and this picture is from a recent summer party, and it illustrates that the weather in Liverpool is much better than its reputation, because this was the beginning of October in sunshine, and you can see people running on the terrace in socks, so it's not too bad. So I'm grateful to work in this team. I would like to thank our collaborators, our funding sources, and you for your time.
Thank you, Christine. We've got time for one quick question. I've got a quick question. Oh, there's a mic about to come.
Christine, very nice. Have you ever tried, instead of reactivating the whole virion, just activating one surface protein to see if it triggers that kill effect?
Well, HIV-1 has a single promoter, so once you switch it on, you have not necessarily the entire program is being initiated, but envelope comes at quite the end of the whole replication or transcriptional part. Okay. So it would be very difficult to specifically activate a specific protein because they are all transcriptionally and post-transcriptionally actually coupled.
Okay.
Thank you.
Totally.
Thank you. Thanks again, Christine. Okay, so final speaker, it's Professor Jonathan Ball, who will share his experience and his scars, I believe, in the development of antibody biologics against emerging viral pathogens. Thank you.
Thank you, Jean. And I've now got two timers. I've got one on my phone, but there was one down here, which is a genius invention. So I'm going to give a whistle-stop tour on some work we've done trying to derive bovine monoclonal antibodies against what are a large collection of pandemic threat agents. I'm going to focus on a target called sarbecoviruses, but I would like to flag at the outset that actually the work initiated was really targeting filoviruses, so things like Ebola virus, but also arenaviruses, particularly Lassa fever. But I'm just going to highlight our progress and our challenges using sarbecos as the target. So we knew some time before the SARS-2 coronavirus outbreak that there were a large collection of viruses out there in bats which had the immediate potential to spill over into humans. In 2002, we had the SARS outbreak. That was relatively easy to contain because it was a virus that caused obvious symptoms. Therefore, you could isolate people who are infected, and you could very quickly bring the infection to an end. However, in 2015, Ralph Barrick had shown that there was this large collection of what we now call sarbecoviruses, which are really primed, ready to go in humans. And here you can see the collection. Here's all of the coronavirus kind of family, and here you've got a collection of viruses which are pretty much here, ready to go into humans. Most of them live in bats. So here you have infection of those, just one example of those sarbecoviruses. You can see the human infection or infection of human cells. You can see the bat virus here has very similar growth kinetics to the human virus, and that is without any adaptation. So if that virus finds its way into humans, then there's a chance that it could go. So how could we potentially fight the next pandemic? Well, we've heard a lot about health infrastructure, surveillance, all of those things that are going to be crucial. However, we do need to be aware that the next pandemic will happen. I am a pessimist by nature. and therefore what can we do to try and be prepared for that? So I think what the SARS-2 outbreak really showed us was that yes, we can call on small molecules, particularly if they've been isolated for other indications. So dexamethasone, for example, treated the symptoms, and it was very effective. So you could pull that off the shelf, have very rapid clinical trials. But if you're developing an antiviral small molecule, that's far more challenging. You're likely to have high attrition, and therefore it's unlikely in the kind of timescale that you want. We saw in SARS-2 clearly the vaccine development, particularly with the modern vaccine platforms, that was very successful. But again, there's quite a significant lag between target identification and development of that vaccine. And one thing that we can do potentially is start to develop an armory of therapeutic antibodies. So these have a very rapid development time, particularly with the technologies that we have these days. And one of the real problems with antibodies is the fact that they are very exquisitely specific. That's why they're good. They don't often have off-target activity, but that exquisite specificity means that very often their cross-reactivity is very narrow, so that you have to develop a new antibody each time you have a new target. So to try and identify an antibody that targets all of these different strains of virus is a challenge. And here you can see, in terms of the antibodies that we developed for SARS-2, you can see that very quickly, with the emergence, particularly the Omicron variants, that all of these antibodies that were developed and heralded as being wonder drugs, when they came onto market, very quickly had marked reduction in their activity against these variants. So the question is, can we develop an antibody that maintains that breadth but still has that specificity? And so to remind you, the challenge that we took on was really thinking about the future pandemic threat of these bat coronaviruses. And so we went in search of a Sarbeca virus monoclonal antibody, and we particularly focused on cows because, Carlsberg don't make antibodies, but if they did, they would be cow antibodies. They're very unusual animals, and they generate antibody diversity not by using lots of different segments that they bolt together. They generate antibodies which have particularly large complementary determining region 3 at CDR3s. Basically, this is the loopy bit that interacts with the target. And this big extended structure here is great at penetrating into pockets. And viruses have evolved to hide a lot of their functionally constrained epitopes, receptor binding domains, fusion regions, occluded in these pockets. Yet these antibodies are able to penetrate deep down inside. So we had two cows that we'd been immunizing with Lassa fever and Ebola antigens, but we were able to then quickly change that immunization to include some SARS spike or SARS-2 spike proteins. And you can see on the left-hand side, the cows had quite a few doses of different versions of the spike protein over an extended period of time. And then what we were able to do, so this is Jack and Dougal, honestly, nothing to do with Father Ted. So we were able to take the blood from those two cows, isolate the PBMCs, do some flow cytometry. It's very hard working with cows because, unfortunately, all the reagents that we're used to having for human antibodies, human B cells, they just don't exist for cows. So this was particularly challenging. This should have been trivial, but it wasn't. So anyway, we sought single B cells. We're then able to do one of two things. If we have single B cells, we can take them through, generate antibodies using normal expression, and then characterize as antibodies. Or we can take them through this route, put them onto the surface of phage, and isolate antibodies that way. So we came up with a panel of spike binding proteins. We got some which had fairly short CDR3s, but we also had these ones with ultra long CDR3s. These are different variants of SARS-2. Green is good, and hopefully you can see just by this table that the ultra long antibodies generally had more green than the others. In other words, they had better cross-reactivity and they were basically more potent. If we took the best of those antibodies, this was an ultra-long antibody that we've called P7, we could show, and this was in collaboration with University of Liverpool, that this antibody prevents hamsters or protects hamsters from SARS challenge. And in fact, this was administered after challenge, so it actually treats hamsters and prevents weight loss, but also accumulation of viral load in the respiratory airway. And that antibody not only protects hamsters, but it also shows potent broad reactivity. So these are different strains of SARS-2, and you can see good reactivity. But you can see here, this black line here, that's the original SARS coronavirus of 2002. So it's working across different sarbecoviruses. And then to finally analyze that antibody, when we looked at a large panel of sarbecoviruses, so these are viruses that have been circulating in bats, we can see P7 has maintained good cross-reactivity against every sarbecovirus that we've reacted with it. So we do believe that we have an antibody with good, potent pan-sarbecovirus activity. Just to acknowledge all the people that did the work. So this was mainly whilst I was at Nottingham. We're now hopefully going to continue this at LSTM with the E&I unit, hopefully in LifeArc, doing some humanisation and then maybe taking it down the clinical pathway. We're also bringing the Lassa antibodies here for the same thing. But I would like to flag Rich, who might be in the audience. There he is. So he was very pivotal in us getting this cow work going. And so please talk to him after. He will tell you how difficult it was compared to working with human B cells. Thank you, Chef.